Hi, everyone, and welcome to the OHBM Open Science Room. We're very excited to have you here, and we're very excited to welcome our global community, everyone joining us from the OHBM virtual platform, and also from Crowdcast and our streaming platforms that we're using to give all of this content to our global community. So today we're starting with day one of the Open Science Room, and today is all about open data. And for this session, we're going to have some lightning talks. So just quick few links on the slide. You can see the OHBM OSR website, and there you can find more information on the schedule and our speakers, and you can look up their bios while they're talking. We also would love for you to share what you experience here on social media. So we have our Twitter name there, and we also have some Twitter hashtags that you can use. If you want to start chatting about our content or afterwards, you can join Mattermost, the Brainac workspace, and we have specific channels there that you can join to join the discussion.
So today, as Stefan said, it's all about open data 2.0. And here we're thinking about the next generation of data sharing. So in this session, we'll have 5 talks of 5 minutes each, followed by 20 minutes of Q&A, where all speakers will be together for you to have their questions answered. Some, but probably not all speakers will be available for live Q&A in this time zone. But any questions that you have for speakers that you can see their talks for now, we'll endeavour to get your questions over to them some way or another.
So we'll be using Crowdcast as our main platform for Q&A. You can use the text-based function there to ask questions and interact with other participants. And you can also upvote these questions, and we'll get them to the speakers in the Q&A session. If you're not using Crowdcast, you can use the OHBM platform chat function or Mattermost to add your questions. And we will add some live feedback polls in Crowdcast as well. And we would love for you to fill those in to get feedback on how we can improve or to give nice feedback to the speakers. And then lastly, we also would like for everyone to stick to the code of conduct. We would want everyone to treat each other with respect and kindness. We all agreed to the code of conduct when we registered for this event. So if you see or experience any violations of this code of conduct, we ask you to follow the specific reporting structure in the code of conduct, and that is detailed on the OHBM website.
So the speakers we'll be hearing from today, first we'll have Emma Bloomkey discussing privacy preserving technology and tools for safe open data use, followed by David Keeter, who will be discussing concept annotations via end-in terms for improved search of open data sets, followed by Linda Larson-Pryor, who will be leveraging telehealth in underserved populations for open data research. Then Julie Boyle will be presenting the Courtois project on neural modelling with their first data release. And finally, we'll have an update from Guiomar Niso on the BIDS community standard. So we hope you enjoy these talks, and we look forward to speaking to you in the Q&A.
Hi, I'm Emma. I'm a PhD student at Oxford and a member of the OpenMind open source community. So in open science, we often talk about data sharing and open data sets. And that's an awesome solution, especially if the data is about bacteria, plants, animals, or space. But we know that we need to be a lot more careful with human data, because you often can't really anonymize a data set. It's more than just your name that can identify you. So about half the US population is likely identifiable by just their gender, date of birth, and their city. And also, data sets can be combined and linked with other public data sets, making them much more easy to de-anonymize. For more information about that, you can search linkage attacks. So what if you remove age, sex, and location? Well, it's also unclear what kind of information can be extracted from seemingly bland data. So for example, you can predict age and sex from even just a structural brain image. You can also be identified by your heartbeat. And of course, look at DNA phenotyping, where even your face can be inferred from your genetic profile. And the thing is, once your data is out there, you can't get it back, right? You can't change your genetic profile or heartbeat like you can a hacked password or credit card. So as you can see, the data that we want to do science on is sometimes actually the same data that can identify you. So stripping it to anonymize sometimes isn't enough. And just in case you're wondering, who's even trying to hack healthcare data? Who even cares? This website has some fascinating graphics, and 41 million health records were stolen in 2019 alone. And just this week, Babylon's health data leak was exposed here in the UK. So for a lot of reasons, not all data can or should be put into a big open access dataset, which is unfortunate. But there are some popular privacy preserving techniques that we can use instead and still do our science. So the first is federated learning. This allows us to train on data that is stored elsewhere, so a hospital, a phone, or another university's server. Instead of copying the data and moving it to us, we can send the model and train there. And then we only send back the weight updates, not the personal data. This can be repeated across datasets in various hospitals or servers to build up a large dataset. This solves one problem, because the data doesn't need to be copied and sent everywhere just to perform computations, training, or stats on it. And that's a big step forward. But sometimes models can memorize details about the data that they've trained on, and they can leak those the details later on. So in a famous inversion attack, for example, they reconstructed a face from the training data. And another famous example probed a text predictor to expose social insurance numbers in the dataset. So this is where other techniques can be handy. One is differential privacy. Differential privacy is a framework that allows us to make formal mathematical guarantees around privacy preservation. So for example, it defines what perfect privacy would be. So in a simplified example, if I have my data set DN and perform a query such as average age, perfect privacy would be if the answer to my query didn't change if one subject were to be removed. So for example, in a data set DN-1. So to come closer to this concept of better privacy, there are many methods that you can use, and one is adding a controlled amount of statistical noise to obscure the data contributions from individuals. And as you can probably guess, this comes down to a trade-off between so-called perfect privacy and keeping your data actually useful, because too much noise can make your data meaningless. And finally, encrypted computation allows computations to be done on data while it remains encrypted. So for more on this, you can Google homomorphic encryption or secure multi-party computation. So if you're worried about how on earth you're going to learn how to do these, don't worry. OpenMind has made free open source libraries that largely take care of a lot of the details for you. So PySift, for example, extends PyTorch, which I'm sure a lot of you have used, so that you can perform operations on tensors. So for example, train or test a model or calculate some statistics without ever moving the data and in a privacy preserving way. And you can do it in Python from your Jupyter notebook. So could these tools be useful for you? Well, yeah, maybe. First, if you're looking for more data. And if you're using deep learning, this is probably the first reason that you thought of. But more exciting is to have more realistic data. We've all seen news stories come out where another company has had an AI healthcare flop as soon as they tried it on clinical data. So Google Health had this. This is a famous example from IBM Watson. And we want to build tools for real patients, right? Not hypothetical patients. But some of us do research, not necessarily clinical stuff. But even then, our subjects are often young white university students, and this just doesn't reflect the world's population or our main patient populations. So if we can use data from other institutions worldwide, we can properly diversify our data sets, and that can help us ensure that our research better serves the world's population and our patient population, because we want our science to reflect the actual population. And finally, another reason why these might be useful for you is if you want to compare your results, perhaps at conferences or to other papers. So for example, with these tools, I could contact a group and quickly test my model on their data set without ever needing to request copies of their data. If you want to learn more, there are a lot of tutorials, for example, on GitHub, there are code demos on the blog. You can really just have a go. And if you need any help with anything, join slack.openmind.org and we can answer your questions. So finally, I'd like to thank all the core contributors and members of the OpenMind community. And for the record, they would love to work with you guys at OHBM and help demonstrate and improve their tools so that they're useful for neuroimaging researchers. Thank you.
Hello, my name is David Keeter, and I'm from the University of California, Irvine. And today I'll be telling you a little bit about the work we've been doing in the NIDM Terms Grant to improve our ability to search across open neuro datasets. This work has been funded under the Brain Initiatives Grant from NIMH and in collaboration with the P41 from NIBIB for ReproNIM, and of course support from the INCF. And see our poster 1973 at OHBM for a little bit more information. So NIDM, or the Neuroimaging Data Model, is a linked data format for storing metadata about human neuroimaging experiments. There's three components: NIDM experiment, NIDM workflows and results. Experiment describes metadata surrounding the experiment collection. Workflows describes the workflows that are applied to that experimental data, and results are for the results. The NIDM Terms Grant, in particular, aims to develop the community-driven controlled vocabulary needed to annotate our data sets and facilitate query across them. We are focused on the open neuro use case and our ability to query across open neuro and apply these techniques more broadly. So in the following image on the left, you'll see this is our workflow that we've gone through to annotate open neuro data sets. The open neuro database on the left, we pull BIDS formatted data sets via data lab from open neuro. You can see in the middle a typical BIDS directory structure that contains tab separated values files, or these TSV files, and optional JSON sidecar files. The JSON sidecar files are meant to sort of describe the variables that you find in the TSV files. Using our NIDM, PyNIDM API, we have a tool, BIDS MRI to NIDM, which iterates over the variables found in the TSV file and interactively queries the interlex, which brings in lots of terminologies, including cognitive atlas, NIDM terminology, BIDS, and so forth. Allows you to annotate your variables with very specific properties, such as units and mins and maxes, but then also lets you selectively associate concepts with those variables, such that things that people might want to use when querying across data sets. So things like a general concept of age. Doesn't matter what units at this level that the variables were measured in, and but the broad concept of age. So then we take these augmented JSON files, feed them back into the BIDS directory structure for inclusion in Open Neuro, but then we also use them to create these NIDM representations that we can use to query across data sets. On this slide, you'll see on the left typical BIDS directory structure, and in the center in red, you'll see a box. This is our augmented BIDS JSON sidecar file. You'll see lots of familiar properties like description, label, levels, and things, but I want to point out one specific property, the isAbout property, which is this link between a variable, in this case gender, which was coded as male and female in the original data set. It is about this broad concept of gender, which just talks about what gender is measuring. So these JSON files are the ones that get fed back into the BIDS data set. We work on the JSON LD version of these, that's the linked data version of the JSON documents, and facilitate our conversion to NIDM. In this little animation here, this is a binder query demo. There's a link at the end of the presentation. Here you can see us scrolling through all the concepts that we've associated with data sets in Open Neuro. Here we're deciding, let's do a query that find data sets that have a measure of anxiety disorder. We don't care exactly at this level how they measured it, but it has a measure of anxiety disorder and it has a measure of age. And we do this query, and we'll find that there are 3 such data sets in Open Neuro that have a measure of anxiety disorder and a measure of age. But I think we need sex too, because we want to do males, age, and anxiety disorder. So we run that query, and we find only 2 of those data sets have variables that measure things about anxiety disorder, age, and sex. The ReproNIM effort has stood up a graph-based database called the Repro Lake, where we're uploading these NIDM metadata files. They contain just the metadata about these data sets. The Repro Lake contains a broad range of metadata files, some from Open Neuro, Abide, ADHD 200, the core data set, These NIDM files can then be used, again, to query across these data sets, and they have links out to the various image files, data sets, and so forth, where you could then go off and go to the source providers and get the actual data or run computation on the cloud. Here are a list of a variety of links that you can explore. I want to highlight the NIDM terms binder query demo. You can do that and run that and play with it. And also this reprint NIDM webinar, which is an hour-long webinar about how to use the annotation tools to annotate your data sets. So I'd like to thank the NIDM terms and reprint teams and the Open Science Steering Committee for selecting our abstract for this lightning talk. Thank you.
I am Linda Larson Pryor, a faculty member at the University of Arkansas for Medical Sciences, or UAMS. I will be speaking to you today about our project to leverage telehealth in underserved populations for open data research. The focus of this study is Parkinson's disease, which is a progressive neurodegenerative disease of aging, resulting in significant impairments in motor, autonomic, and cognitive function. In Arkansas, a largely rural state, 3 of the 4 movement disorders trained neurologists practice at the Movement Disorders Clinic at UAMS, with about 75% of the clinic's 756 patients residing in medically underserved areas, or MUAs, noted in orange on this state map. Motor and cognitive impairments limit the ability of many Parkinson's patients to drive, making them reliant on family and community members to take them to and from medical appointments, and limiting their ability to participate in research. Those limitations can result in reduced quality of care, while the inability to be part of research programs adds to our recruitment biases. There is a recognized need to address the known variability in human populations and an increased awareness of the need to share data collections and results across laboratories to provide meaningful data on such phenotypically variable data as that presented in Parkinson's disease. To address these issues, we have established a multidisciplinary team of informaticists, digital health experts, clinicians, and imaging scientists. Our project leverages a new neurology telehealth program focused on Parkinson's that is being developed by a collaboration between the Movement Disorders Clinic in Neurology and the Institute for Digital Health Innovations. Our research assessments that are noted here in purple will be accomplished at scheduled telehealth neurology appointments, with patients provided the opportunity to take part in the study at the time they schedule their telehealth appointment. We will collect self-report information on sleep, cognitive function, and motor freezing via REDCap, capture speech as an audio file, handwriting and finger movements over smartphone or via a computer interface, or if necessary, on paper. These data will be digitally acquired and submitted to the Arkansas Image Enterprise System, or ARIES, from multiple sources noted here. Research data will be de-identified at collection, and a unique study identifier linked to that individual's clinical record at the Arkansas Clinical Data Repository via a newly developed tool by the Data Repository team. Imaging data will be collected from the UAMS Picture Archiving and Communication System and passed to POSDA, the Perl open source DICOM archive system for data anonymization, quality control, and tracking. We will leverage the capabilities of the ARIES platform for semantic integration of diverse data sets to remove the obstacles in working with different source representations for similar types of information, enabling the connection and interpretation of different data types concerning the same phenomenon and diverse data that are about the same individuals. This figure illustrates the basic approach for the use of ontologies to integrate diverse multimodal data into a semantic repository, showing how these data would actually be stored as a resource description framework, or RDF, in a triple store, which is a type of graph database. The ovals in this diagram depict ontology classes. The small circles are instances, and the arrows are the relationships between these elements. In blue, I have represented the linkages between neuroimaging and cognitive assessments in a specific individual subject noted here of the type Homo sapiens. Here you can see the study identifier linked to the same individual. The result is a combined graph that links together all the information we have about each subject, and this instance data is aligned with ontology classes that contain a lot of background information that isn't pictured here. Our goal is to improve the quality of care for underserved regions of Arkansas by increasing access to care and enabling individuals in underserved areas for whom travel is an obstacle to participate in research and feel empowered by being part of the research enterprise. We hope to provide a blueprint for other researchers to gather large and less biased data from human subjects. We will provide one mechanism to accomplish this by creating a semantically integrated data repository that is findable, accessible, interoperable, and reusable, in which fully de-identified data in the ARIES collection may easily be made openly available to the research community.
Hi, my name is Julie Boyle. I'm the project manager for the Courtois project on neuronal modeling, also called Neuromod. And today I'm going to talk to you about our first data release. So one of the main aims of the Courtois Neuromod project is to train artificial neural networks to behave in a more human-like manner. And we think we can do this by achieving, this can be achieved by using extensive whole brain activity recordings of brain networks in naturalistic tasks. And in order to be able to train the artificial neural nets, we plan on acquiring an extensive amount of neuroimaging data on a small number of participants. And of course, once we have that data, we'd like to openly share it with the community, the scientific community, which is what I'm here to talk to you about today. So just to give you an overview of the project, the Courtois Neuromod project scans 6 healthy individuals, 3 women, 3 men. We plan on scanning each of the participants 100 hours per year, so that's 500 hours total over the duration of the project. And we've been scanning participants on a wide range of cognitive tasks that I'll talk to you a little bit about in a second. Participants are scanned both in fMRI and MEG, and once participants arrive at the scanner, they go in with their customized CaseForge head cases, and then we hook them up to an MRI-compatible biopack system, so we can acquire biosignals that include cardiac signals like ECG and pulse, respiration, skin conductance, and we also have a high-resolution eye tracking system. So for 2020, we plan on releasing 3 fMRI data sets that accumulate 31 hours of data. That includes movie 10, so that's 12 hours of movie watching in the scanner. Participants watch Born Supremacy, Wolf of Wall Street, Hidden Figures, and Life, and they watch Hidden Figures and Life twice. They also watched the entire first season of Friends, and we did a task in the scanner that we're calling HCP test retest, where 4 participants did 15 repetitions of the HCP functional localizer. Now, it's all fun to talk about data, but we're here to tell you how to get data. So the first thing I suggest is that you check out docs.cneuromod.ca. That's our documentation website, where you can find all sorts of details about the tasks themselves and scanning parameters. We plan on having a beta release arriving this summer. We were hoping to have it in time for OHBM, but like everyone else, COVID delayed us. We hope to have a release candidate in September 2020, so that's gonna be a more polished version of the database, and hopefully with all the bugs out of it by then. So in order to be able to get access to this data, you have to go through a process that's called registered access. So the PI of your lab has to use their university credentials, write a short blurb about the type of research you plan on doing. And then once that's done, you get approved, our institutions will sign a data transfer agreement, and the data is yours. So data is organized in BIDS formatting, and we'll be using a DataLad. The advantage of using DataLad is that the structure and the types of files that are included in the data release are going to be public. So you'll be able to see what is in our data release before you actually have gone through the registered access. But if you click on one of the files to be able to download it, you're only going to be able to do so if you've gone through the registered access and you have a unique identifier and password. So things to look forward to. In 2021, we've already started acquiring some data for the next data release, which is going to include playing video games in the scanner. So we've done this with a couple of participants where they've played Shinobi 3 in the scanner. Image 10,000, which is what it sounds like. It's 10,000 images, so they're naturalistic images, of which you can see 2 examples on the screen, where they've been curated to be high quality and in the public domain. You can expect more seasons of Friends. We love Friends at Neuromod. an emotions task where participants watch short video clips that have been annotated for their emotional content, a language triplet task that you have an example of here on the screen, and a memory task. So finish up, I'd like to take a second to give a special thanks to Elizabeth and Arnaud, who have been helping us with the data release, Tammy and Anwar, who at the beginning were super kind to give us advice on how to acquire movie watching in the scanner, and most importantly, the pitfalls to avoid. And of course, the whole Neuromod team. And if you want more information, we have a Twitter account, a GitHub account, and a cneuromod.ca website that we encourage everyone to check out. So thank you very much.
Hi, in this lightning talk, I'm going to present the BITS, a data standard to support the neuroimaging community. Maybe some of you have heard about BITS, especially these days. BITS, the brain imaging data structure, is a standard to store, organize, and share your neuroimaging data. Why is it needed? This could be a potential situation in a lab. This is Professor Smith. Hey, I need to ask Mike about this old data. There's so much more we can do with it. But this is Mike. He doesn't longer work in the lab. So Professor Smith needs a lot of help. Why is that? Data is organized differently between institutions and even within a lab. So this produces suboptimal uses of resources, human resources with a lot of time waste trying to rearrange the data, infrastructure with data storage space, and financial because this organized data has very limited value after the first publication. On top of that, the results are very poorly reproduced, even within the lab where the data were collected. So BITS, it's a standard for neuroimaging data, try to solve all this. What is BITS? BITS is based on very simple file formats and folder structures. BITS is not a file format, but it specifies which file formats to use, for example, Nifty, JSON, or TSV files. It also specifies the naming convention for files and directories, and which is very important too. It prevents your metadata getting lost while doing the research. Metadata is stored in JSON files, which are readable by both humans and machines. The principles behind BITS, the first one is adoption, which is crucial. We try to minimize complexity and facilitate adoption. We don't want to reinvent the wheel. We want to reuse existing methods and technologies whenever possible. We also follow try to tackle 80% of the most common used neuroimaging data derivatives and models, and engage the global neuroimaging community. This is a BITS dataset example. This is your dataset, and you will have a participant file, a dataset description, then your subject organized by session and imaging modality, your anatomical MRI, functional MRI, diffusion, or in another session maybe you have MEG, behavioral, and so on. So for example, your participant TSV is just a very simple file that includes some keywords and some description. So this is really easy to adopt and requires very minimum energy. BITS is a community-led standard, which is the result of ongoing collaboration of amazing people. And the specification is created after sharing knowledge, discussions, and consensus through the email discussion list, through the shared Google Docs, and through GitHub. We have a GitHub page with a lot of collaborations and contributors. We now have a very well-specified governance structure. We also have some BITS extensions which are called webs, and each of them has a leader which is in charge of the coordination. And we have a BITS steering group This is our current BITS steering group, and it's made by people all over the world, from around the world, and each of us with expertise in different imaging modalities. And we also have a really nice team of BITS maintainers. And on top of that, and the most important, is that BITS is a very open and inclusive community with hundreds of contributors around the world. And I really encourage you are very welcome to join and contribute whenever you feel it. And we have a starter kit for people that doesn't know how to approach, and a BITS validator, which is a tool to just check your datasets it's following the BITS standard and it's compliant with the standard. Yeah. So the BITS originated at Stanford in 2016, and very soon got extended to many other imaging modalities such as MEG, AEG, intracranial EEG, task events, electrophysiology, behavioral, and very recently genetics. So many modalities, and many more to come. We have a lot of extensions which are work in progress now, and like PET, CT, very recently added NIRS, and we also have derivatives. We are currently on the last round of the Guiomar derivatives before adding them to the standard in an official way. So I really encourage you, if you want to give any feedback, to go to GitHub and just provide any comment you feel like. So organizing your data in BITS is good for future users, and it's good for future you. Thank you very much. I really encourage you you are very welcome to join the BITS community, and I also encourage you to attend the BITS Open Science Room emerging session that will take place very soon. Thank you very much.